Right, so I'll try to keep this short. I'm Ben, my partner Brian couldn't be here, but we had this project trying to fine-tune the epigenetic clock. So the main thing about what we were trying to do is that there are a lot of age-related diseases, and they generally occur as people get older and older, and those are generally very featured in epigenetics. And the biggest measured thing of this has been previously DNA methylation, which has been quantifiably linked by Horvath, who measured that using a simple linear regression model that there was a big link between DNA methylation and biological age. So we wanted to basically put a spin on that and basically do the same thing, but using histone modifications, using 4 histone modifications. We've searched papers, and there seems to be a large impact on age. Some of these impacts are contradictory in some papers, different in terms of model organisms. We wanted to see if we could put together a link between these. So the first thing we did is trying to find features that we can use to build a machine learning model. So in the epigenetics, the ChIP-seq experiments find the proteins of the histone modifications to the DNA, so you get a signal over the entire epigenome. And we could have used this raw signal, but our first experiments, like 2 terabytes of data for just small sample types. So we just used the number of peaks, and functionally that might have been much better because peaks are the more important areas generally in terms of expression. And the important thing is not just predicting age, but from these machine learning models, we want to see the interpretability of these. So in a linear model, a larger rate would imply that that region is more important, judging by the difference in the counts of peaks in that area. So the largest sample that we had was whole blood, which had the most number of samples across multiple histone marks. We use the IHEC data portal, which used different experiments such as ENCODE, roadmap, European data. But the age ranges were very varied. Ages in 50 to 60, 50 to 70 were very, very dense, whereas younger ages were not. So that was by far the largest challenge of this. And unfortunately, we didn't get very good results at first, especially with some of the more complicated models, which we thought would do much better. So the simple linear models actually seem to do the best across most of the data types. We also tried using a polynomial support vector machine with degrees 2 and 3, because we thought that might capture combinatorial features of different areas of the genome, and random forest, which might prevent different types of, just as a different approach. And we looked at using neural nets to try to capture higher order functions. In this case, we just multi-layer perceptron, we would give them convolutional recurrent networks. These were all optimized hyperparameters and measured on the mean absolute error. So with this, we basically just went with the linear regression model. Looking at the actual data, though, it didn't seem to be as promising as we thought it was. As seen on the plot, there's a very high bias in the model, and the R-squared was not very good on this. This is most likely due to the number of samples. So we started balancing and found an optimal balancing by cutting off limiting the top 80% of each bin. Because if we strictly balanced everything, it would absolutely destroy every mean absolute error in our squared that we found. And another thing we wanted to do is, because these are histone modifications and not just cytosine methylations, we wanted to explore combinatorial effects. We thought this would be a much better approach, but the problem with this is, again, the the number of data points especially, and samples were not consistent across these. So it was very hard to produce a better model with that, even though we also tried different approaches such as binning similar ages. Finally, we wanted to use some of the weights and better features that we found from the linear regression models to try to make predictions on what these found. Although none of these were significant, there were quite a few different functional annotations that we found that looked to be important in organismal health or development. So that's the whole presentation.
So I'm curious, why did you use a number instead of the total intensity of a genomic marks in a particular region? Because the peak caller itself might just be calling a bunch of little things or one big one, but that's just a peak caller feature. If you instead looked at what is the total number of reads, basically what is the integral under that curve, what is the total intensity? Yeah,
so we looked at using the median, the max, the min.
Initially, the sum of
the sum as well.
The sum of all reads of all things.
Of all just total signal of all the reads in the region.
Yeah.
We did that initially on K27AC.
Yeah.
We got similar mean absolute error. Unfortunately, I think the big roadblock was just the amount of data. Just for 400 samples, it was terabytes of data, and it took a long time to train and get that data. So I think that looking into that with more complicated models might.
So basically, what you're looking for is regions of the genome whose total H2K27 isolation call is the data.
Yes.
Why is that so much data? Can't you just bin the genome and look at?
Oh, it was downloading the entire read files and all that.
Got it.
Taking all that data.
Yeah. And then the other aspect to think about, can you go back to your marks that you looked at?
Yeah.
K27, okay. So K27 trimethyl is an interesting one. Basically, we found in general that regressive marks are more correlated with age.
Yeah, we found that in the literature as well. So which is surprising because we didn't find it.
Yeah, that's interesting. I'm curious if it's the way you're measuring it or something else.
Yeah, but we didn't actually do we didn't do the number of reads for H2K27 methylation.
You should try that.
Yeah, that might be interesting to look at.
Yeah. And did you try the inflammation as a DNA methylation as one more feature, just to sort of as a sanity check to make sure that the stuff that we know works also does work?
Yeah, we didn't try that. It's been published
just a positive control. So, I know it's been published, but basically knowing that you're going to apply the same method to something that works and actually find a result that works would reassure me that there's no bug, for example.
I see.
Like in the way that you're processing the data or analyzing.
Okay.
Does that make sense?
Yeah. Cool.
Other questions?
How did you do the recon?
So that was all pre-generated. I think it was just max 2 from all the ones.
All right, that's it.